Last time we had already gone through this actually, but I thought this would be a decent place to start. And what we're going to focus on mostly right today is essentially right here. Okay, we're going to talk about this part right here. And that is that there's a group of proteins, and I'll show you a picture of it so it'll be more clear, in just a minute or two. But there are proteins that, and you see this that looks like the lollipop here with the yellow. These proteins are actually in the mitochondrial outer membrane. They're embedded in the membrane. And here's one called BCL2. There's another one, BCLXL, which is in the same family. And what these things do is essentially prevent the death of the cell. And so we're going to go past this stuff that we looked at before. And we talked about this last time, that there's whole groups of proteins that are both responsible for life of the cell and the death of a cell. And then this is actually where we left off, right? We were talking about all of these proteins. And so the prototypical member is BCL2. And BCL2 is a pro-survival protein. It has a transmembrane portion, which is what this TM stands for. This actually has domains that are named after itself. So it's a little weird. It's not named after something else. It's a BH3 homology, like a BCL2 homology region, 1, 2, 3, and 4. And there are other proteins that are related. In fact, there's a lot of them. So this one called BAX, it also is a transmembrane protein. If you see this one, you know that it can stick inside the membrane. It has homology here with these 1, 2, and 3. And then BH4, it does not have a similar region. And then there are some of them like BID, and there's actually a whole bunch of them that have what are called BH3 only, right? BH3 only proteins, because the only way that they're homologous to this one is through this region that's colored red. They have a similar region. And the key to this is not to memorize the numbers or names or groups or anything. The key is that these homology regions allow these proteins to interact. And it's the interactions of these proteins that controls whether or not a cell is going to live or die. Okay? So don't memorize the names. Don't worry about how many domains there are. Realize that the survival of the cell is dependent on the ability of these things to interact and either allow death to occur or prevent that from happening. Okay, so again, some of them are anti-apoptotic, some are pro-apoptotic, some of them are these BH3 onlys, and actually that's probably the biggest group, and we'll look at some of them in a bit. But what happens is it takes two to tango, as I say. It's always about interactions. Right? They don't do much by themselves. It's groups. So these BCL2 family, right? That includes all of those on the previous slide. That doesn't mean that they're pro-survival. The BCL2 family is all of those proteins because they're homologous. They're similar to each other. Okay. So these proteins form homodimers and heterodimers. Homo means two of the same protein. Hetero means two different. Mm-hmm. And they interact with each other. And the release of cytochrome C, which we saw in that awe-inspiring video that I'm sure you shared with everyone that you know, right? The release of cytochrome C from the mitochondria is dependent on the formation of a pore, right? I mean, you can't get out unless there's a tube to let you out. Exactly. Yes. And the pore formation is dependent on the pro-apoptotic proteins like BAX and BAC to move to the mitochondrial membrane after activation and form a pore. Okay, So BAC is constitutively in the mitochondrial membrane. Constitutively is just the fancy way of saying all the time, right? It's just that's where it's present. And so BAC is there, but BAX is actually, after it's activated, after an oncogene or there's DNA damage or something, and P53 activates it, that gene, that's going to be produced and it's going to go into the mitochondrial membrane where it can interact, right? BAX and BAC, because it forms heterodimers. These two can then interact and form a pore, a channel, an opening in the membrane that allows the cytochrome C to get out. When the cytochrome C gets out, it binds to other proteins and it forms that apoptosome that we talked about and allows the cell to die. Okay?
So BAX is not in the membrane. It goes to the membrane where BAC is always, and then it like activates it and they form the pore.
Yeah. Oh. It interacts with it. It moves to the membrane after it is activated. It inserts into the membrane. Okay. And then also on the membrane, because I said this is a battle, we have BCL2 and BCLXL that you saw in that last slide. And they essentially picture them as sponges or interceptors. Their job is to bind to the pro-death things and prevent them from interacting with each other. Right? So if the two of you sticking to each other, right? I mean, I can only see two people. So there's the two of you, right? So if you stuck to him, right, if that caused the death, right? If there was a third person there that could grab him instead and he couldn't hold you, then we're going to prevent that from happening. Does that make sense?
Yes.
And that's exactly what happens, right? So this decision of death of the cell is based on relative amounts of these groups of proteins. Does that make sense?
Yes.
So if I have a lot of BCL2, which is keeping the cell alive, then the cell isn't going to die. If we have a lot of BACs and BAC, then it's going to overwhelm those defenses and you're going to get death. And that's actually what happens with oncogenes. This is how cancer cells are recognized, is that there's a threshold of BACs or BAC that are turned on, and it essentially overwhelms the defenses and allows the cell to die. In a cancer cell that's successful, that doesn't die, right, that avoids apoptosis, they're going to have to either turn down the pro-death signals, right, prevent that from happening by, for instance, destroying the P53 pathway, or they're going to have to turn up the defenses, right, so that no matter what P53 throws at them, they can intercept that and stop it from interacting. Okay? And so this is what that looks like. Okay, this is from a paper from 10 years ago where they were figuring this out. In this system, they overexpressed. They had tons of BCL2 in their system, and they could turn it on and off. And what they found is that as soon as you turn off expression of BCL2, the cells would die. And what that was telling them was that the cancer, the oncogene, was constantly sending death signals, die, 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 die. But this large amount of BCL2 was sucking up all those signals. And it's all because they can interact with each other in a bunch of different ways. That's the take-home message, right? Is these things are interacting in a bunch of different ways with each other. So here's an example of what would happen in a mitochondrion. If you have a situation where you have a lot of BCL2, okay, so here, this is the BCL2, this yellowish looking moon lollipop thing, right?
Right.
Okay. And the BH3 only proteins are sent in to activate BACs or BAC. But if there's a lot of BCL2 there, BCL2 has a very high affinity. It binds very well to these things, and it essentially soaks them up. Right? They're more likely to bind to BCL2 than they are to back or backs.
Mm-hmm.
If the amount of, and you see they have it, I don't know if you can see it on your screen, there's just a little shadow there. So if you remove the BCL2, so it doesn't have to be entirely gone, but if the BCL2 levels go down, then these BH3 only proteins can bind to backs and back, and that allows them to interact and form a pore. So it's these heterodimers, and then this, of course, is an oligomer. There's more than two. There's a bunch of them. They come together, and they essentially poke a hole in that mitochondrial membrane and allow the cytochrome C to get out. When the cytochrome C gets out, it binds to APAF1, forms an apoptosome, and the cell's a goner. Okay, does that make sense?
Yes.
Are you good?
Yes.
Okay. So this is the critical thing, right? So these are just some examples of them. Sorry, I'm moving you all over the screen here. Just try to so it doesn't block the slide. So there's a bunch of anti-apoptotic proteins. Again, memorization is pointless, right? There's a bunch of them, and this isn't all of them. The only one that you should remember, if you want to remember one, is probably BCL2, because that is the prototype, right? That's the one that all of the others are based on. And the pro-apoptotic members come in a couple categories. There's those that have 2 or 3 of the BH3 member, and then there's the ones that are BH3 only.
Mm-hmm.
Okay, so there's backs and back that have lots of members. There's a bunch of BH3. Okay, hold on. Let's see. Yalda's coming. All right. So Yalda, I gotta hang up on you and then add you into the other talk. Okay.
Okay.
You know these proteins, right? That there's these pro-apoptotic proteins and anti-apoptotic proteins. And so a very good question that you might have is, why are there so many of them, right? I mean, you look at this and you think, how many? You only need one to kill a cell, right? So it seems crazy to have a dozen of these things like spike and harikari and all this stuff, right? It seems really funny. So why are there so many BH3 proteins? Okay, and these are essentially those BH3 only proteins. And
only?
Hello?
No, it's fine.
You can see.
Yes.
Okay, this is amazing. Okay, this is the first time. We should write this down. It actually worked. I don't know why my thing kept dying, but it did. Okay, so the issue is that if you're a cell, then you have multiple things that you need to be aware of that may be unrelated physically inside the cell that could trigger death. And so I'll just give you some examples. So if you have DNA damage, or if you have low oxygen levels, then the cell will trigger P53, and you're going to then go through particular death signals, which are going to block BCL2, right, and the related proteins. But it may be that what you are lacking is a cytokine. Maybe there's a particular growth factor or something that keeps this cell alive that's missing. Or in this case, Taxol, the chemotherapy drug up here, Taxol right here, works by disrupting microtubules. So if you're a cell, you may want to check on your DNA, but you also have to know whether or not the cytoskeleton is in good shape, and whether or not you're holding on to the extracellular matrix. Are you attached? I mentioned this word one time. I said that when cells are not attached to something, be it the extracellular matrix or a plate if you're growing it in culture, if you release a cell, it'll die by a mechanism called anoikis. And that essentially says the cell says, I'm no longer holding on to anything. Goodbye, cruel world. Okay? And it is this particular BH3 only protein that will do that. And so if we look at it this way, right, and we kind of close up, BIM is a BH3 only protein. It is normally sequestered by being bound to the motor complex on microtubules. You're aware of dynin, right? The motor molecule on microtubules, and UV exposure causes the phosphorylation of BIM. It gets released from this motor molecule and goes in. So this is a way of the cell saying, we've likely been damaged. All right. Here's another one. This one binds to another motor complex. Again, UV irradiation or release from the substrate or the extracellular matrix. If the cell's floating, it causes it to get released. That's going to go over and interact with BCL2. But the anti-apoptosis players, that is, the ones that are blocking death, also are affiliated with things. In this case, AKT, that pro-survival protein we mentioned in the beginning of the class when I was talking about oncogenes, it phosphorylates bad. That leads this BIM, this BH3 only protein, to be sequestered. Okay, so what you have here is a very tight integrated signaling. And as I told you, it's a battle, it's a dance, it's about balance. So if the overwhelming signal is we have lots of pro-death things coming in, right? We have DNA damage, we're turning on a bunch of these, the blue ones are the pro-death signals, right? And then they're going to be combated, but we can overwhelm the defenses. That's essentially what has to happen to kill a cell. All right? So normal cell, the situation is that it's alive and it's being kept alive. If you can overwhelm those defenses with pro-death signals, you can cause the release from the mitochondria and the activation of the caspases and death. All right? Alternatively, you activate them with external signals like we talked about, something coming in from a T cell or another immune cell. Does that make sense to everyone? Mm-hmm. Right? So it's a battle. It's not a one signal fits all kind of thing, right? You have multiple signals being integrated, and ultimately the cell is essentially like a scale. The cell is essentially saying, well, in a normal situation, I have signals that are keeping me afloat, alive. And there's probably always some signals that are causing this same cell to want to die, but they balance out, right? All of the death signals are able to be intercepted by the life signals. And then when a cell has become sensitized, essentially what that means is there's an increase in pro-death signaling. Probably because oncogenes are activated, DNA is damaged, right? The cell is in trouble. That would be considered sensitized. But the cell then eventually, the pro-life signals start to go away, and just like hanging weights on this balloon, the threshold will be met where there's enough death signals to outweigh essentially the force of pro-life, and the cell's gonna die, right? And so in cancer, the things that keep cancer cells alive is an increase in the pro-life signals, an increase in BCL2 and its friends, and interference with the pro-death signals like P53. Does that make sense to everybody? And just to give you an example, I don't even know if you can see it. I know I'm much too old to see this at this small. But if you look at the big picture, as I call it, you have lots of pro-death signals, lots of pro-life signals, and it's too complicated really to just summarize. But our take-home message, again, the bottom line, right, is that it's a balance of the pro-life proteins interacting with pro-death proteins that ultimately makes the decision for a cell. Okay. So it's the balance of life and death that controls the growth of a tumor. So if you have a tumor cell or collection of cells that has a low division rate and it also has a low death rate, it's not going to grow very quickly. If you have one that has a high division rate, but it's also dying very quickly because the threshold is quickly being met, you also don't have a lot of growth. The cancers that tend to be really problematic are the ones where you have a high division rate and the death rate is low. Either because the pro-life proteins are produced at too high a rate, or because the pro-death proteins, like those controlled by P53, are no longer active. Does that make sense to everybody?
Yeah.
Okay.
P53 fails, then the division can overcome them.
Say I'm sorry. Just repeat that, Marina.
That P53 is failing, that the division rate overwhelms the death rate, and then you have a problematic.
Absolutely, yeah, because P53 directly turns on BAX and directly turns on the genes that cause cells to die. So if you lose P53, you're going to lose those pro-death signals. Right? And then the cells are going to survive when they shouldn't. Okay. So what would you expect to see if you look in cancer, right? I mean, what we care about are patients. And so what do you expect to see? You expect to see overexpression of the anti-apoptotic genes, right? That would help a cancer cell. And it turns out that the BCL2, the founder of these proteins, was actually identified in a lymphoma, right? I mean, this is a protein, the gene that was identified as a breakpoint in a translocation in a B-cell lymphoma. So BCL2 is actually named after a cancer. It stands for B-cell lymphoma. Okay. So what happened in this case was there was a breakpoint. The BCL2 was no longer regulated the way it was supposed to. It was expressed at very high levels that overwhelmed any kind of death signals, and this cell was able to live when it shouldn't. Okay? The inhibitor of apoptosis proteins, IAP proteins, these are going to be expressed at high levels. Right? And you would expect a loss of expression, or at least a loss of activity, of the pro-apoptotic genes like BAX and BAC. You would completely expect that, right? It turns out that some viruses even encode proteins that look like BCL2 that reduce apoptosis. They're able to defend themselves. Right, because viral infection is something that triggers cell death, but the viruses like EBV that cause cancer are able to overcome that, right, by producing proteins that mimic the activity of BCL2. Right, and we already mentioned this, so I won't go over that. Some cancers can actually interfere with the extrinsic mechanism of generating death by interfering with the Fas ligand. They act like that. They bind to the Fas protein, but they don't activate the death receptor. So essentially, they send out decoys, right, that prevent the cells from dying when they should. Okay. Now, is this something that can be used to help patients, right? This seems like very deep inside the cell, and it seems like maybe it wouldn't help a patient. But it turns out that this is something that's actively being studied. So if you remember the diagram I showed you, actually before you got on, Yalda, but on the mitochondrial membrane, there is this BCL2 proteins, and what people have done is make BH3 mimetic. A mimetic is something that mimics or looks like something else. So this thing acts like a BH3 only protein. It's called ABT737. ABT is the drug company, Abbott. Okay. So what do you think? What would this drug do?
It would make it compete with the actual cytotoxic stresses.
So if this thing acts like this protein, how is that going to affect? Is that going to cause cells to die or not die?
It would promote apoptosis.
Yeah, absolutely. Mariana, you got that?
Yes.
So this thing acts like a BH3 only. It gets complicated because it's a negative regulator of something that's a negative regulator. So it interferes with BCL2, but BCL2 is blocking death. Right? So indirectly, this thing is turning on these guys and allowing the permeabilization, the release of the cytochrome and caspases and death. Okay, and this is what it looks like. If you look at it, the structure, if you're familiar, you see all these amino groups. You see that it looks like a protein, right, with side chains. Okay, it's not exactly a protein, but it's a peptidomimetic. It looks like this thing that this BCL2 binds to. It looks like a protein. Okay. And here is an example of the way this stuff works. Hypoxic human cancer cells sensitized to this BH3 mimetic induced apoptosis because it downregulates BCL2 protein MCL1. MCL1 and BCL2 are similar, right? It's pro-life. And so these things are able to actually induce death, right? Here's the compound, ABT737. Right. And here is a search for clinical trials of this drug, right? So this is a bunch of a bunch of recruiting trials, completed trials. So this drug, all of these treatments are designed to interfere with the apoptosis, with BCL2 and the BCL2-like proteins. Right? So there are now a bunch of drugs that are designed to either... Yeah.
You're breaking up.
With who? My God. Or is it with whom? Anyway.
Okay, no, it's better.
Yeah. Okay. So, but no treatment is without side effects, right? So all drugs have side effects, and so this is an example I thought I would show you that when you antagonize an inhibitor of apoptosis, i.e., you are increasing apoptosis, right, with a chemical, what they found was that it actually led to increased bone metastasis, which is a terrible outcome of a cancer treatment. And so here is what happens when you treat the animals with a vehicle, i.e. just saline. And here's what happens when you use this inhibitor it's an inhibitor of apoptosis. I'm sorry that it's so complicated, but you get the idea. If you inhibit an inhibitor, you're helping something. So this BV6 is something that causes apoptosis in the cancer cells, but look at what happens to the bones, right? They're eroded, right? Compared to this one. So this is something that they clearly have to be aware of, right? When you're trying to develop a treatment, you have to be aware that you may be causing unwanted effects. Okay. Okay. Are we good on all of that? Yalda, the person behind you is raising their hand. I don't know if they have questions or not.
They're studying anatomy.
Oh, okay. She was just very engaged. I don't know. Okay. All right. So are we good with all of that?
Yes.
Yes.
Marina, you're good? Yeah, because I can only see the person who's talking at the time. So every time somebody talks, it switches. So Yalda, I can watch her cough because she made the most recent noise. Okay. And I can watch those people study anatomy. I'll tell you if they do anything interesting.
Okay.
Yeah. All right. So the next thing that we're going to talk about, we've talked now about tumor suppressors, about oncogenes. We talked about the signals that control death in normal cells, but in particular in cancer cells. And almost all of the treatments that we're going to talk about later, almost every treatment works by triggering death via apoptosis. Okay, so apoptosis is not just something that's sort of academic thing to understand. It's the way the treatments work, right? So the better we understand it, the better we're able to trigger the death in cancer cells and not in regular cells, in healthy cells. So what we're going to talk about now and for much of the rest of the class is, in fact, the way that the tumor cells interact with other cells, right, with their environment. They don't act alone. And in fact, the environment is extremely important in the development of the cancer. So now we're going to switch and we're going to talk about the tumor microenvironment, the interaction of the tumor with its host cells. Okay, and I sent you this PowerPoint as well.
Thank you.
I don't know if you've gotten time to see it, Yalda. I'm sure you didn't, and Mariana, and I don't know. I don't know that you would have seen it either, but you have it, and I'll post both the video and you'll have the PowerPoint. Okay.
Thank you.
If you don't get it, let me know because it was a pretty big file.
Okay.
So it may get blocked, and I'll have to shrink it down or something.
Okay.
Okay. So up until this point, right, how did we get where we are in the understanding that we have in the class, right? So much of the work that was done until this point was based on single-celled models, right? So either yeast or bacteria or using cancer cells that were grown in culture, but it was still monoculture, right? We took the bladder cancer and we grew that in a dish. Right, we saw that, or we separated these things out.
So they model them to figure out like the metabolism and the reactions.
Yeah, I mean the reason that it was so important to separate the cancer cells from everything else back then was we didn't have the tools to look at complex systems, first of all. But if you're going to isolate oncogenes, it makes sense to separate the cancer cells from everything else. And that's what they did. And when people studied cancer, really up until the last maybe even 10 years, I mean, it's not been the history. We're talking thousands of years people looked at cancer, but it's only been a decade or so that people have really been able to look beyond the cancer cell itself, right? Once we understood about oncogenes and tumor suppressors, that worked really well. The systems of breaking open cells and getting out their DNA and having one cell type, it was great. But it really doesn't work well in real life, right? Looking at these genes in isolation, you're looking at the cancer cells in a dish, does not mimic what we see when we look in people, right? Tumors are very complicated, and to understand why treatments work or don't work is really important that we look at the whole system. And so that's what we're going to look at now. So what is in a tumor? Well, the cancer cells, which a lot of people probably, I would think, Mariana, and so when you guys go out in the community and talk with people, when people think of a tumor, they think of a blob of cancer cells, okay? And there's much more to it. The cancer cells make up only a small portion of a tumor, and it can be even 1% or less would be the actual carcinoma cells, those transformed cells. And many tumors, I'm going to turn it down a little bit. And then it sounded like there was a plane landing in one of your hallways or something. You'll tell me if you can't hear me. But a lot of tumors, almost all of the cells are non-cancer cells. And the general term for all of the cells that are in a tumor that beside the cancer cells are the stromal cells. Right? Stromal. Right? And they just refer to the whole thing as the stroma. Right? The stroma is everything else. Right? Stromal refers to the situation. Stroma, you would say, oh, they're in the stroma. Those are the stromal cells. Got it? And there's a very large amount of extracellular matrix. I'll actually show you a picture in a couple minutes and you can see what it looks like. But it's the reason, for instance, that you can detect tumors by feeling them is because there's so much matrix. So here is an example of a tumor. You have your carcinoma cells. Here they are, the big pink ones, right, in the bottom. But you have epithelial cells that are not cancer cells, right? So if you have a tumor in a breast, a lot of the cells are normal, right? Thank you for writing. You have the cancer cells. You have this thin layer that separates the tissues called the basement membrane. And that surrounds all of our organs. It's a fibrous, thin, kind of not transparent, but a very thin, kind of milky clear thing called the basement membrane. You have the extracellular matrix that is produced by the cells in the area. But then you have a bunch of other cell types too. You have the endothelium. These are the cells that make up the blood vessels and lymphatic vessels. Endo means within. Endo, within these tubes, right? But within the blood vessels and lymphatic vessels. There are lots of fibroblasts. And fibroblasts are one of the principal proteins that secrete the extracellular matrix. The principal cells that secrete the proteins of the ECM. Okay? And then you have immune cells, right? Be they macrophages, NK cells, lymphocytes, T cells, B cells, right? Neutrophils. There's a bunch of mast cells. There's lots and lots of different immune cells that can be inside a tumor. Okay. And the cells in a tumor, sorry, I'm hesitating only because like the screen's like flashing back and forth. Can everyone see everything?
Yes.
Okay. So the cells in a tumor talk to each other all the time. So here's a list of the other cells present. The fibroblasts. They make extracellular matrix proteins. They produce growth factors, such as fibroblast-derived growth factor, or FGF. There are cells that are called myo. Myo, what does that make you think of? Myosin, right? What is that related to?
Myosin protein.
Right. And what does myosin make you think of?
In muscles.
Muscles, right? So myofibroblasts are contractile things. They make actin. These are the proteins that surround blood vessels and help squeeze them. They help to regulate blood pressure. Right? They squeeze those vessels and release that pressure. The myofibroblasts are the ones that are going to surround the blood vessels and lymphatic vessels. The endothelial cells are the cells that actually make up the blood vessels or lymphatic vessels, right? They make growth factors. They form these tubes that allow for movement. I don't know why this is acting so weird. The pericytes are cells that are located near blood vessels that support the blood vessel function. It turns out that pericytes are really important for response to chemotherapy. There's smooth muscle cells inside a tumor that line blood vessels that help with contraction. There's adipocytes, which forms are adipose is fat. So adipocytes are cells that store fat. They're similar in structure to fibroblasts. And then there are lots of immune cells. Okay, and we'll talk about that in a little bit more. But one thing to remember, and if you're following the news lately, you know that there's a lot of information out there now about the microbiome. What is that?
Microbiome is like the composition of microorganisms.
Yeah, exactly. I mean, it's all of the microorganisms that are there. So we now know that the microbiomes, the microbiota, right? These list of hundreds or thousands of different kinds of bacteria absolutely controls inflammation, interacts with the immune system. So here is a picture of a diagram. Here we have a tumor that arises in animals that have a microbiome that is pro-inflammatory. Here you have the gut unraveled sort of in animals that don't have that flora, and they essentially have no cancer. Right? Everyone got that? So we now know that that microbiome is critical in colon cancer. It's very likely to be important in skin cancer and other cancers as well, right? Even your eyes, which you don't typically think about it, your eyes are covered with bacteria. You have an eye microbiome.
And amoeba?
Not amoeba, hopefully, but you have lots of bacteria and probably...
I saw that amoeba could go into your eyes and cause from the cornea.
Oh yeah. Who was it? Who was it that just got that? There was a woman that just got.
Yeah.
Yeah, I'm trying to think of it. I remember seeing.
They said it was from like contaminated water.
Yeah, but it was a girl who left her contacts in.
Yeah.
Forever, and her contacts were disgusting, and she got an infection, and it ate her eyeball essentially. Yeah, that's what it was. But I mean, that's not a normal flora, of course, because she was disgusting and left her contact lenses in forever. I mean, like half a year or something.
Well,
yeah. But here's an interesting one that shows you about how the composition of a tumor is very dynamic, and that your behavior, even things that you don't really think about, can really influence stuff. Fragmented sleep. So that is people that slept only in little bits and woke up all the time. It actually accelerates tumor growth and progression because it alters the immune cells that are inside the tumor. Mariana, did you say something?
Yes, that was lost in the last part. He said that people that get fragmented sleep, and then we hear anything.
Sorry, it's just loud because of all the people in the background in the two places. Okay. So what is it like inside a tumor? Okay, you guys can still see, right? Because I really can't see you anymore for some reason.
Yes, we can see.
Now I see you. Briefly. Okay. So what is it like inside a tumor? The first thing is that there are low oxygen levels. The blood vessels that form inside a tumor, and we haven't talked about angiogenesis yet, but the blood vessels that form inside a tumor are abnormal. You just can take my word for it now. And they don't really distribute oxygen well. and so it's very hypoxic. There's low levels of oxygen. And when you have hypoxia, when you have low levels of oxygen, the cells will metabolize the sugar that's there, and they can't continue into aerobic respiration because there's not enough oxygen. So they will convert that into acid, right? Pyruvic acid, lactic acid, etc. And so the inside of a tumor tends to become acidic, right? A decreased pH. Right? And one of the things that will happen when you have an acid environment and you do not have enough oxygen is that the tumors will start to die in the middle. And I think I showed you a picture of that previously, of a sort of rotting, disgusting tumor in a dog.
Yeah.
Okay? And that's pretty common, right? That the center of a large tumor will become necrotic. And so again, necrosis is not like apoptosis. It's messy. The cells die. They spill their guts. It can lead to inflammation by releasing all the stuff inside the cell. You can have a reaction of the body to this necrosis. And what we're going to talk about when we talk about metastasis is that when you have hypoxia and acidosis, if you were a cell, what would you try to do? What would happen to you as a human if I put you into a hypoxic, acidic environment? What would you do?
Try to get out.
You would try to get out, right? You would get the hell out of there, right? And that's exactly what happens with the cancer cells. So this environment, this hypoxic, acidotic, necrotic environment, right, which is shown in this diagram, right? The cells will try to get out of there. And that's one of the major driving causes of metastasis, right? Is the cells actually trying to survive. So if you look at this is a dye that this thing is staining for hypoxia and oxygen levels. Here's the capillary. You have an area that surrounds it, but you see it's not very big, in which you have normal oxygen levels. Then there is an area of hypoxia, low oxygen, and then you have this black ring here where these cells are not getting enough oxygen to live. And so they undergo necrosis. Right? But in this area here where you have hypoxia, these are the cells that are going to want to be getting out of Dodge, right? Because they're in a very bad environment. Does everybody get that?
Yes.
Yeah.
Okay. So these are the ones that we're going to have to worry about when it comes to metastasis. Right? These guys are dead. These guys are happy. It's these that we have to worry about. Now, what else is going on in there? Because the blood vessels in the tumors are very abnormal, they're leaky, and they don't get really good blood flow because they're very bendy. They twist and turn a lot, so it's hard to get good flow. You have low blood flow, and the dividing cancer cells put pressure on the vessels and squeeze them. So it's very hard to get things in or out of a tumor. So what tends to happen is that the pressure in the area around the tumor goes up. The fluid leaks out, but there's not a way to get rid of it. Normal stuff, what happens is the fluid leaves our blood vessels and it gets absorbed back into the lymphatic vessels, and then ultimately gets dumped back into circulation in your neck, right? There's a junction between those. And the lymphatic fluid gets dumped back into the blood. But that doesn't happen in tumors because the lymphatic system is inadequate to do that. So you get this leaking fluid out into the tissue. You get a swelling in there, and you get increased pressure. All right. The structure inside a tumor is chaotic. It is disorganized. It doesn't look like a normal tissue, and I'll show you some pictures of that as we go. But you have essentially disorganized extracellular matrix, and the cells are laying every which way. So there was someone named Otto Warburg, and years ago, he said that cancer, above all other diseases, has countless secondary causes. But even for cancer, there is only one prime cause. And he said that essentially it was alteration of the respiration. So instead of burning up oxygen in respiration, there was fermentation. And of course, he was to at least partly right. Okay, so cancer cells do undergo what's called aerobic glycolysis. And what that means is that even in the presence of oxygen, because the human itself, the person, has plenty of oxygen, the cancer cells will preferentially get their energy from glycolysis. They don't use respiration. They don't use the Krebs cycle. They don't use the mitochondria to produce their ATP. And so this is the basis of one of a very common imaging test for cancer. Have you ever heard of PET scans?
Sounds, but I don't really know how it works.
Right? So a PET imaging, it stands for positron emission tomography. I don't know if I have a picture. No, I don't. I should have a picture of a PET scan. But in the PET scan, what happens is you infuse the person, you inject them with a form of glucose that is radioactive. It has a radioactive fluorine. It's fluorodeoxyglucose. Right? It has fluorine in it. And this radioactive glucose accumulates in the areas where the tumor is because the tumors are using so much sugar. And then when you look at where the radiation is inside the person, you can visualize that with a color when the radiation is detected. And PET scans will essentially have bright spots that show you cells that are using lots of glucose. Does that make sense? And so PET scans, unlike CAT scans, right? You can use a CAT scan, which is like a bunch of x-rays. The CAT scan tells you structure. The PET scan tells you whether something is actively metabolizing or not. So you can see something on a CAT scan. It doesn't tell you whether that thing is growing or not. It just tells you that there's something there. PET scan actually tells you whether or not something is actively metabolizing sugar and actively metabolic. And so sometimes they'll be combined. People can do PET CTs to see, hey, there's a blob there. Is it actively metabolizing sugar, right? Is it growing? And it's thought that the acidity that's generated by the tumor microenvironment actually helps to drive invasion. Because for the reasons that Mariana said, right, that these cancer cells have acidity, there's low pH, there's low oxygen, they essentially want to get out of there.
Yeah.
I'm yawning, probably because I can't see you, because I'm talking to a blue square. Yeah, it's not as much fun.
Do you want me to turn my video on?
Do you have your video off? Is that what's going on here?
No, I thought it would help your internet speed. Okay. Do you see me now?
I do see you.
Okay.
And I still see the girl. Yeah. Okay, so the key to this whole thing, much easier to teach someone if I see them. The key to this whole thing, think how weird it is. I'm sitting here talking to a computer, right? So the communication is key. And so I thought I'm going to briefly go over this. This is something you probably, because the people in ITESM are essentially black shadows because they're in the dark. So they just look evil. So the communication is key. So I thought I would just define some terms for you because these are things that you're going to see. You probably already know these, but I thought I would just A, to have it in English, and B, just to refresh it quickly. There's autocrine signaling. Autocrine is essentially, auto means self, right?
Yes.
And so an autocrine signal is something that's produced by a cell that acts on that same cell, which sounds weird, but the immune cells do that all the time. T cells produce things that act on other T cells and on themselves. It's a way of quickly getting a reaction. And it's not uncommon. It seems weird. Juxtacrine is the next level away, right? If we go from self, the next thing would be to work on someone who's touching you. And the term for that is juxtacrine. If you have ever heard of the term juxtaposed, that means that two things are touching. Okay? So juxtacrine means that the signal goes directly from one cell to the other. We talked about this already when we talked about the extrinsic death pathway. You can have a cell like a T cell that touches a cancer cell and causes that cancer cell to die. Right? By engaging that death receptor on that cell. Paracrine signal is nearby. So these are affecting local things, paracrine signaling. A good example of paracrine signaling is nitric oxide, right? That gets all the attention because it causes men to get erections. And so nitric oxide is produced locally. It acts only on cells very close by. It has a very short half-life. This is destroyed within a fraction of a second. So it doesn't get to diffuse very far in order to give its signal. Does everyone get that?
Yes.
Right? It acts only nearby. And then there's endocrine signaling, which is produced in one organ or tissue, such as a hormone that would come from the brain, and actually goes through the body and gets a response somewhere else, like the gut. Are we good?
Yes.
Okay. In cancer, every one of these is important, right? Whether you're talking about the insulin that's produced in the pancreas that's going somewhere, whether you're talking about growth factors that are controlling either the growth of the cancer cell, or maybe this is a blood vessel that we're regulating. Juxtacrine signaling, that is the cancer cell talking to an immune cell or the cancer cell talking to a fibroblast. All of these things matter. How do you become independent of growth signals? Maybe you're making your own growth signal, autocrine signaling. Okay? So all of these immediately are relevant to cancer. Okay? Now, immune cells are important enough that I separated them into... Geraldo, you got to tell those people to be quiet. They're having too much fun back there. Just go over and slap her. She'll really be surprised. She can't even hear me, right? Because I'm talking into your headphones?
Yes.
Right. So if you just went over and just whacked her in the side of the head, I bet she'd really be surprised. I bet she'd be quiet. At least for a minute. I don't know. You're tall. She's not bigger than you, is she? Right? She's Asian looking. She's probably only like 5'2" or something. You can take her. Yeah. All right, so immune cells. Immune cells are really important both because they're responsible for eliminating the cancer cells, but actually we're really understanding now that immune cells are important because they're causing problems, right? Again, this is probably about the 5th or 6th time that I've done this. I put it in bold. This should be ingrained in your brains. Inflammation and cancer are tightly intertwined.
Yeah.
Right? Whether it's inflammation because you're obese, which causes inflammatory response in the prostate or somewhere else, or yes.
So in that article that you provided for that nematode worm.
Yes, nice. Nematode.
Nematode. So they said that they actually anything that could mimic that inflammatory path could cause cancer.
Yeah.
And the reason that of course, one of the reasons, I mean, the most important reason that we have carcinogen that still are unknown is that because anything could.
Yeah, that's actually a great question. And I don't know if they heard you or not. So her question, can you hear her?
Yes.
Wow, what a great software. So her question is like, is the reason that we don't know everything that causes cancer because there are so many things that may cause chronic inflammation or inflammation that we don't know about? And that's it. The answer has to be yes. Right? I mean, many of the known carcinogens, we know them because we know they cause DNA changes, right? That's the most common test is the Ames test for mutagenesis. That's what we can test. But how do you test chronic inflammation? Right? That's a lot harder to test. What are you going to put it in an animal and wait 30 years? You know?
Testing for inflammation?
Yeah, so it's just a much harder thing to do, especially on an isolated chemical. Certainly we know that in humans, we now know that things like Crohn's disease or irritable bowel syndrome or even asbestos, which we know causes cancer, right? Causes mesothelioma, the lung cancer it's associated with in healing it. It doesn't cause DNA damage. It causes chronic inflammation. Right? So as far as we know, everything from hepatitis virus to asbestos, which is a crystal, to a nematode worm that can cause chronic inflammation, so far everything we've looked at that does that is also going to be a carcinogen. Okay? And what controls inflammation in a large part is the immune system. And so that's why we're going to spend some time talking about that. Okay? So macrophages, macro means big, phage means eater. These are cells that ingest a lot of other things. They regulate the immune response by presenting antigens to immune cells. They produce a lot of cytokines, a lot of small molecule, little proteins that regulate the activity and division of other immune cells. The immune system is its own world, right? It's an entire army, a whole military with all kinds of, it's got a navy, it's got air force, it's got everything. So it's very complicated. We're going to skim over it. I have no choice but to do that. But if you really want to understand cancer and the new treatments, you're going to have to dig deeper after this class. Okay? So macrophages. This is what macrophages look like. Here they are interacting with other bacteria. They produce growth factors. They produce proteolytic enzymes. These are protein slicing enzymes. Macrophages are really important in tissue remodeling. That is, they chop up tissues and allow them to reform. This is important whether you're recovering from an infection, right? Whether you have a cut and you want to heal that cut, you have to allow blood vessels to come in. The macrophages are going to cut the path for those blood vessels to be able to come in. Does that make sense? So anytime you're breaking up and building down tissue, they're involved in this. They're able to induce angiogenesis, that is blood vessel formation, and they may kill tumor cells directly. Lymphocytes. Lympho refers to the fact that these live in the lymphatic system. The main ones are the T cells and B cells, T and B lymphocytes. Cyte just means cell, so lympho, living cell. They release chemokines and growth factors. The T cells are able to kill cancer cells directly by that extrinsic pathway. Tell me, Alda, if you're going to beat them up because I want to be able to watch the video. How many are there?
3.
3. That's nothing. There's nothing. I mean, the first one will be so surprised. The other 2, you know. Yeah. So then you have granulocytes, right? Granules. They have a lot of grains in them. Oh, look, they're packing up. Yeah. They have a lot of granules in them that contain enzymes, that contain cytokines that they release. And so that includes reactive oxygen species or enzymes that produce them, like superoxide dismutase and others, and nitrogen metabolites like nitric oxide, right, which itself is a nitrogen metabolite. So the granulocytes, so when you have a chronic infection like hepatitis, right? Remember we talked about these viruses that cause cancer. If you have a chronic infection that's causing chronic inflammation and release of these reactive oxygen species, the cells in that tissue are going to be dying. If you don't want to lose the organ altogether, you're going to have to make more of them. The liver, in particular, is very good at reproducing, regenerating. So when you have a viral infection, I take it back. They look like they're loading for, she looks like she's loading up a pen or pencil or something. She's doing something. They're not going anywhere. So, see, this is just like one of those CIA movies. I can tell you what to do and where to go. Head to your left. Yeah. So these reactive oxygen species cause the cells to die, but the cells have to reproduce. So picture yourself now. You are in a liver. You are being damaged by a virus. You have reactive oxygen species that cause mutations. That's how they kill targets, right? They damage membranes. They damage DNA. And now you're reproducing over and over again in the presence of those agents. Do you see a recipe for disaster here? Right? You have a terrible mixture of DNA damage, lots of replication, and over again for a long period of time. Okay. Mast cells, which are here, right? This is what mast cells look like. They're full of little grains. Mast cells, typically when you think of a mast cell, you're thinking of allergic reactions. So when you go outside and you have a terrible reaction to pollen, right, nowadays, right, especially in the fall, there's terrible pollen here. What happens is that the mast cells will bind to antibody like IgE, an antibody, and they degranulate. They release a lot of histamines and things that cause your blood vessels to dilate and become leaky, and that's why you start to drip and everything swells up. So they cause swelling. Mast cells are, in fact, though, related to cancer. NK cells are called, they get their name natural killer cells. Natural killer cells. And these NK cells are thought to constantly patrol the body. Their other name is, they call them large granular lymphocyte, or LGL. So they look like lymphocytes, they have a great big nucleus, and they're able to directly bind to cancer cells and kill them. Okay? Now, during the development of cancer, right, from its inception, the initiation of cancer, through the neoplastic, new growth, neoplasm, new growth, through this development of this new growth, this development of a cancer, you have constantly pro-inflammatory cytokines that are being produced. You have the innate immune system producing chemicals like the neutrophils, the granulocytes producing things that are causing tissue expansion and conversion. The immune system seems to be intimately involved. And of course, that sounds like a terrible idea. And of course, it is a terrible idea, right? The immune system isn't doing it on purpose. It's responding in a normal way to stimulation. Right? The problem is when that stimulation is long-term, right? Whether that means it's because you're obese and you have these cytokines that are being produced in your pancreas and prostate and tissue and breast for 20 years, or whether it's because you have some kind of long-term infection like hepatitis virus, and so you have long-term inflammation, or whether you have inhaled asbestos and you have inflammation around that area in your lungs for 20 years. Right? Right. Hola. Are you okay, Mariana? You guys getting in trouble or something?
Sorry.
Are you okay there?
Yes.
Okay, how are we doing on time?
I think it's almost time to finish. Yes, we have like 5 more minutes or so.
Okay, well, I mean, actually. Okay, yeah, I'll just actually do this one slide and then we'll go. Or you can ask questions. And Yalda can just sit on for an extra 15 minutes because she was late. So
Okay.
she can just stare at this slide the whole time. Okay, so a lot of cancers are associated with inflammation, and we've talked about a lot of these: hepatitis, the Helicobacter, the bacteria that's associated with stomach cancer, Crohn's disease, ulcerative colitis, etc., with colon cancer, gingivitis, with oral cancers. It is also known that the opposite is true, that if you treat someone with non-steroidal anti-inflammatory drugs, something like just aspirin, right? Or the COX, which is a COX-1 inhibitor, by the way, or a cyclooxygenase, that's what COX stands for, a cyclooxygenase COX-2 inhibitor, something like the next generation, like the Motrin and those guys, right? Vioxx, these things that are, they're leaving now, y'all, to say goodbye. Or at least she left. I can only see one.
Yeah, they all do.
Yeah. The COX-2 inhibitors, like I take actually 81 milligram aspirin, a low dose aspirin every day.
Yeah.
One reason is because it seems to help with cardiovascular disease.
Yeah.
The other is that it seems to reduce colon cancer risk. So NSAIDs and COX-2 inhibitors actually do reduce it. There's examples, like for instance, this in a mouse model system where they were expressing the human papillomavirus, the oncogene, the early region genes. If you have a system that doesn't have mast cells, you can reduce cancer levels, right? And this has been around a long time. I actually showed you this picture before and told you this when we talked about the history of cancer, right? That Virchow, the father of the cellular theory of life, right? Cells coming from pre-existing cells. He actually said that he thought there was a link between cancer and inflammation in the 1800s. Right? And everything that we know now very strongly supports the role of inflammation, in particular chronic inflammation, in the development of cancer. So I think this is probably as good a time to stop as any. If you have any questions, I'm happy to answer them, and then I'll let you go, except Yalda, who has to be held behind after class because she was late.
Yeah. I have a question, but I think it's because everyone is driving at the offices right now, so now the sound is disturbing them. But I just wanted to ask quickly about the acidic environment. I've heard many people say that diet is related to creating an acidic environment that actually promotes tumor growth. But what you told us today is that the acidic environment makes the cells go to other places, not up here.
Yeah, I mean, so when you see things, and I meant to mention that when you go out in the community, you're going to see things about like, the acidic environment, and people are going to tell you about water that is alkaline water and things like that. Have you heard of that, right? That's supposed to prevent cancer. There is no way to change the pH of your body in general. If you were to do that, you would die. In fact, there's all kinds of systems in your blood to prevent it from changing pH, right? So you'll see on the internet and you'll hear about that sort of local environment you know, the people say, oh, change your pH and eat this food and you'll raise your pH. And the reason we get cancer is because we eat low pH foods, and that's a bunch of crap.
Okay.
You cannot change the pH of your body without causing yourself to get really sick. Okay, so when you're talking just about that microenvironment, the area inside a tumor, it is low pH, and it does actually cause them to change, as we'll talk about later. It's a process called the EMT, but we'll come back to it. I don't want to put it all in and then not explain it, but it does change the behavior of the cells. Like you said, they want to look for a better place to live, and that's what happens.
Thank you.
Yeah. Anything else?
No, not for now.
Okay. Okay. All right. Well, I will see you next week then.
Yes.
And you should